Hello, I'm Elaine Murado. I'm from Loyola University Chicago in the US. And I think we might give folks just a minute or two, since we just opened the webinar, to see who's coming in. This will be taped, so we'll give it a minute. Stephen, would you like to introduce yourself? So we're co-moderating the session, so welcome to our session of Look Who's Talking Now, Capturing Patient and Provider's Perspective in Health Data. I'm co-moderating it with Stephen. You want to?
Sure. Hi, everybody. My name is Stephen Johnston. I'm a Senior Director of Real World Data Analytics and Research in Johnson & Johnson's Medical Device Epidemiology Group. So,
yeah,
pleasure to.
It's our pleasure to be here and have our speakers. It's a wonderful grouping of oral presentations that encompass many of the disciplines that are in health economics or health outcomes research in our ISPE society, as well as drug utilization. And as you look across these talks, we're going to be hearing different ways of approaching patient and provider reported data, whether that be through new technologies like smartwatch or new healthcare delivery modalities like telemedicine and its increased use, and how we best utilize electronic health record data as well. So I get the pleasure of the introduction with a bit of the housekeeping details. What we're asking is all attendees to make sure you're on mute. That includes for those on the panel speakers who are not talking. Questions, though, can be entered into the chat function in the Zoom menu, and please enter your questions at any time during the presentation. What we're going to be using as a format is about 10 minutes of talk and 5 minutes of Q&A, and so we'll be able to answer questions at the end of the presentation. And it's a reminder also that all sessions are being recorded, and they will be available for the meeting after the meeting is over. Okay. So without further ado, I'm going to introduce our first presentation, and it is from Dr. Jeffrey Curtis, who's a professor of medicine, epidemiology, and computer science at the University of Alabama, Birmingham, and will be presenting their work on use of smartwatch technology as one way of collecting patient-reported outcomes in rheumatoid arthritis patients. Thank you.
Thank you very much. Can people see my slides?
Yes, we can.
Wonderful, thank you. Well, on behalf of myself and my co-authors, I appreciate the program committee for inviting us to present the results of our work related to patient engagement in the Arthritis Power Registry, which is one of the 20 PCORI patient-powered research network registries to capture both smartwatch and patient-reported outcome data. And the use case disease that I'm going to focus the presentation on is the study which was conducted in patients with rheumatoid arthritis. I have the following financial relationships that people can read through. This study was an ancillary study to the registry sponsored by Eli Lilly. And as I mentioned, the Arthritis Power Patient-Powered Research Network is one of the 20 patient registries. And so that's part of PCORNet. So we wanted to acknowledge our initial PCORI funding. So by way of background, and the very fact that all of us are here in this session today, I think probably there's great interest amongst all of us in understanding how different types of patient-generated data both reflects patients' experience, but then thinking forward, how might we integrate patient-generated data, including electronically collected patient-reported outcome measures, or ePRO measures, into real-world evidence data sources. There's also, of course, interest because people get tired of answering questions in grabbing passive data, like biometric data from consumer-grade or research-grade health devices that can measure such things. So that's a second aspect of this project. The goal of this analysis and presentation is to characterize the engagement with patients, protocol adherence, and the completeness of data in this study, which now was ongoing but has been recently completed, the Digital Tracking of Arthritis Longitudinally, or digital study, which again is one of now about 40 studies that are nested within the Arthritis Power Registry. So patients were invited to join this app-based study. So the interesting feature about this is an entirely remote study. So nobody's being onboarded at a clinic. There's no doctor engagement. There's no medical center. So this is entirely remote. So that presents some challenges. One of them is that because what we're asking you to do would end up yielding compensation in the form of a consumer grade health tracker device, a Fitbit, we wanted to make sure that people would actually do this study. And so we required a bit of a run-in. That run-in asked people over a 2-week period to give us a little bit of data, to basically just say that you really want to be part of that study. So for the first 10 to 14 days, we collected a numeric rating scale asking one question or one item about pain and fatigue, as well as multiple once weekly electronic PROs. So you had to qualify to do those things with a minimum adherence over that period of time, and I'll describe that in a bit more detail because that's rather central to the presentation. If you didn't complete the run-in and satisfy those requirements at day 14, you were permitted to try again. And the main part of the study was 84 days or 12 weeks. We customized, as we do for all the Arthritis Power studies, we have a customized workflow that I'll just give you a bit of insight into visually. Patients use their own smartphone. We're not buying them a smartphone, but they were mailed a Fitbit Versa if they qualified for the main study. So they gave us a little bit of ePro data up front, and then we added on to that the Versa, which they received at no cost. So for this analysis, we made descriptive comparisons between individuals who entered screening and entered that run-in period and successfully completed it based upon that minimum adherence criteria versus those who didn't. And then secondly, people who were adherent during the 84 days of the study versus those who were non-adherent. This gives you just a little bit of a visual feel for what kinds of screens that people were looking at and they were asked to do. So this was, of course, formatted to fit in an app. We told them on the rightmost panel, how much you have left to go. We use predominantly the computer adaptive testing versions of any of the instruments, including the NIH PROMIS instruments that are part of this study to minimize participant burden. It looks fairly similar during the course of the main study period. On the rightmost side of this, people can see their own data longitudinally over time in the spark lines that shows them trends. So this is the notion of the quantified self, that this is not just one-way information transfer, we're giving people back their own information. So the method schema is described up top here. So there's essentially a week -2, or if needed, -4 before you enter the main study period, and then it's conducted over the next 12 weeks. If people started not to give us data or were falling off a bit in terms of their adherence, we had a hierarchical method to promote adherence. We started with simple things, automated in-app reminders. We then sent them semi-automated custom text messages using SMS or secure text messaging integration with the app. And then if needed, if you didn't respond to automated reminders and then customized reminders, then part of the consent for the study was that a human could call you. That's, of course, the most expensive thing that you could do in any sort of clinical setting or research study. So you save that to last for only the people that need it. We engaged people using those methods with rules-based alerts. So if, for example, you hadn't given us, ePro data for more than 3 days, then this whole process would be initiated and then would escalate by day 5 that a human would call you. Similarly, if you failed to sync the Fitbit data or you just plain old weren't wearing it, the same process. So the human intervention is saved for last so as to be efficient with resources, particularly people's time doing case management or study coordinators. Adherence for this analysis and for the whole study was defined as giving us daily PRO data for more than 70% of the 84 days of the main study, more than 70% of the smartwatch data, weekly ePro data, so that meant you had to give us at least 9 of the 12 weeks of the study. So basically that's as close to 70% as we can get, and then a composite, which was all of the above. So initially, 470 people expressed interest in the study, and a total of 59.1% of them completed the run-in period. So you would say, wow, you have 41% drop-off. Remember, everybody's being onboarded remote. They're not in a doctor's office. But what's quite insightful is the leftmost panel, which compares the people that did not, and the formatting's a bit off, I apologize, that did not complete the run-in period versus those on the right. So just to orient you, on the x-axis, that's the number of days into the run-in period. So starting on the left, by definition, everybody is adherent. That's why there's a solid blue circle. That blue dot is an adherent day. On the next day, it turns pink if people weren't adherent, if that adherence falls below 70%. So every column is the next day into the run-in period of the study. So what you see in the leftmost panel, there's a lot of people that give us ePro data on day one, and then you see the second column is pink, and then it says pink, and forever after is pink. And what that means is people gave us data on day one, and then they actually never give us anything again. So what you're looking for as a pattern is you're looking for, when do those blue dots start becoming pink? Where on the x-axis does that occur? And what you can notice is that for some of this, that occurs on day two, meaning people gave you data for one day and then they dropped off. For some of them, the pattern is they get to about a week and then they start becoming non-adherent, and a few are beyond that, but mostly there's some big gaps at around day 2 and then around day 7. On the rightmost side, those are people that completed the run-in period successfully, and as you would expect, for the most part, they start blue, they start adherent, they stay adherent, but in the top quarter of it, about 25% of people were mostly adherent, and then something happened. You know, by day 13 or 14, it fell below 70%, that mostly unbroken string of blue dots, and every row is a patient here. It turns red. So up here in the rightmost corner, those are the people that asked for a second chance, and we gave them a little bit more grace, and ultimately they qualified, and that's where they're in the rightmost panel. So the implications of this important figure are, if you're going to have a run-in period to promote engagement and make sure people will really do this, how long does it need to be? What we concluded is probably a week would be great, but in fact, it might be able to be shorter, like 2 or 3 days. And if you onboarded them in clinic, probably this is a worst case scenario, and you can likely do even better than that. These are the characteristics of the patients who qualified for the main study versus those who didn't. There aren't huge differences, with only a couple exceptions. One is that the 278 that qualified for the main study were more likely to be employed, 55% versus 39.6%. Those were either homemakers or retired or disabled. They were somewhat more likely to be on biologic and or conventional synthetic DMARD treated. And interestingly, they had somewhat lower pain, and pretty much all of their patient-reported outcomes are a bit lower, but in fact, more similar perhaps to the general rheumatoid arthritis patient. This is the data related to overall adherence now during the main study type, according to what kind of data it is that we're looking at. So what you see is that the best adherence in blue is with once weekly ePro data. So 87% of people were adherent, 82% of people were adherent to wearing the smartwatch. The daily data was a little bit lower at 57%, and if you ended all of those, that was more like 53% adherence. The weekly data and wearing the smartwatch, clearly most people were willing to do that over these 12 weeks. And then finally, we looked at some characteristics of people who met the composite adherence definition versus those who didn't. A few of these are significant. There aren't gigantic differences quantitatively. Those that met the composite adherence definition, so they did everything we asked them to, are a little bit older, 51 versus 48 years, a little bit more likely to be white, 90% versus 80%. Most of these other characteristics, though, are not meaningfully different. And then if we look to see, you know, could you predict or could you foreshadow the likelihood that they would be more or less adherent even during the run-in period or the lead-in period? Again, there's some small differences, but they aren't exceedingly large between these. And we're currently looking at building a prediction model that would help us understand at a patient level who's likely to stay engaged throughout the course of the study, since there are some differences even during the run-in period, as well as early on during the course of the study. So in conclusions, compared to other digital health RA studies, this completely virtual, meaning onboarding was remote, not at a doctor office, not at a clinic or a study visit, was quite good, over 80% for both weekly ePro data as well as the smartwatch data. The potential reasons, we attribute this in some part to the fact that we have this lead-in or run-in period. We think that's an important design element, particularly if you don't have a lot of in-person engagement. We also adopted user-centered design principles and had a patient-centered app workflow that was unique to this particular study. Those with greater pain and fatigue were somewhat less likely to qualify. And although there might be some trade-offs in generalizability, in fact, compared to external referent data, the characteristics of the people that qualified are actually more like the typical rheumatoid arthritis patient. So with that, I'd like to thank you very much for your interest and attention, and field any comments or questions we may have in the time available.
Well, thank you very much. That was a really interesting study. And let me ask if folks have any questions that they'd like to ask, please put in the chat. I have one to start us off. I noticed that you were able to do this work embedded within a large research network, and others may be inspired by what you've presented and would like to start something similarly. In the spirit of the pragmatic advice you were giving, what key considerations should someone be thinking about to make something like this work in their own research or evaluation?
So I think that as people think about the V's of big data, the velocity, the volume, the variety, I would chalk up value and the value of who is your stakeholder audience, what's in it for them? Why will patients want to participate? And I think a lot of researchers design an app or build a custom digital workflow for data capture simply for search, but that doesn't excite or motivate patients. So anything where you can return patients' data back to them, but that you're engaging with humans or helping them derive some of their own health insights from the data you're gathering, ideally if you can connect that to some clinical process or even their own care. But I think thinking about the patient or participant as a stakeholder that is going to want more than just compensation to retain and to remain in the study and remain engaged, those are really important patient-centered user design principles that you need to be attentive to. And when researchers build something only to collect data with one-way information transfer, I think we've had notable examples where we see problems and there's huge attrition over time.
Yes. So really thinking about the value proposition of participating in these data collection beyond just here's some money, but rather folks are, no. I don't see any other questions coming through, so I'm going to ask one more quick one. Did it surprise you to see the decay and the missingness of data over time and how quickly that occurred among people who are motivated to participate? And did that change any of your analytics strategy and how you were thinking about missingness?
No, it really did. And, we learned some of this on the fly. I think if you onboarded people in person, and we're currently doing that with another large study in rheumatoid arthritis, I think the mechanics and the optics might be a little bit different for participants, but it definitely did. There's clearly people that are interested to learn about it, and then they're going to be quite erratic quite quickly. We saw that in that dot plot that I showed, but for the most part, if they're still with you by that point, they're probably likely to stay with you for the whole time. But adjusting to some of these things on the fly was definitely a necessity. So absolutely plan for that, pilot test, beta test this even before you pilot test it, perhaps. That's one component. The other thing that I think was a little bit unanticipated, this needs to be what our team calls grandma proof. Meaning this needs to be so easy for people to do. Everything that you can set up ahead of time and make simple for people, do that, because anything that can go wrong does go wrong. And we all know that in research. But, we played more tech support even with consumer grade devices that you can buy, on Amazon or the local electronics store, much more than we thought. So just be prepared for that.
Yeah. Thank you. We have to be our own geek squad based on you.
That's right. Exactly.
Yeah, I'll pass the baton on now to my co-moderator. Thank you very much, Jeffrey.
Thank you, Elaine. Thank you, Jeffrey. Up next, we have Katja Verheem, who's an associate professor at the Department of Medical Informatics at Erasmus University in the Netherlands, and she'll be presenting on a rapid response drug utilization study using EHR data from 6 European countries.
Hello, everyone, and thank you for the committee to inviting us to present our data. So on behalf of my co-authors, it is our pleasure to show you the results in our study on the use of ranitidine and other H2 receptor antagonists, rapid response drug utilization study. This is my conflict of interest and also of conflict of interest of the co-authors. And for this research, we received an unconditional research grant from the EMA. Some background, ranitidine is an H2 receptor antagonist to reduce stomach acid production. It's a quite old drug. It was launched in 1981. And 2 years ago, there were some preliminary lab analysis showing the presence of NDMA, a human carcinogen in ranitidine. And based on these findings, the regulatory authorities wanted to have more information on use of ranitidine, and they asked us to conduct a drug utilization study to investigate use of ranitidine in real life. And that data was needed to guide the decision whether regulatory actions were needed. It was indeed a short study, which you can see, or a rapid study, which you can see here on this slide, with showing the timelines. So you can see at the right that it took 4.5 months to do these drug utilization activities and go from protocol approval to manuscript drafting. What were the research questions? So we wanted to investigate the prevalence and incidence of use to ranitidine and other H2 receptor antagonists. We wanted to investigate characteristics with regard to age, sex, calendar year, and cumulative duration, which was important because of its potential carcinogenic effects, and then also the indication of use. Now, if we move to the methods, so we use data from 6 primary care databases from 6 countries in Europe, and you see these databases here, and they were mapped to the OMOP Common Data Model. The principle of OMOP Common Data Model you probably know, but it is shown in this figure, and we have electronic healthcare databases with their heterogeneous aspect, but by mapping them to the common data model, they become homogeneous in structure, which allows us to conduct research, pharmacoepidemiologic research in a faster way. Our study period was from 1992 to 2019, which was important because, as I said, ranitidine was launched upon the 80s, so it was important to go back as long as possible. We then selected ranitidine and other H2 receptor antagonists based on RX norm codes. From the prescriptions, we calculated the cumulative duration, and then we focused on the incidence of use. So these were new users, number of new users in a calendar year, assuming that they are under the condition that they had not used in the previous year. And in the denominator, we had a number of subjects with observation time in the calendar year of interest. Prevalence denominator remained the same, but in the nominator, we counted all patients being exposed to either ranitidine or another H2 receptor antagonist in the calendar year. We also focused on the indication of use, which could be, so the indication of interest was either reflux disease, peptic ulcers with or without Helicobacter pylori, or Zollinger-Ellison syndrome, which is a rare syndrome where too much gastric acid is produced. And how did we assess this? We searched for disease codes, in particular SNOMED codes, in the year prior to the first prescription of a subject during follow-up. Covariates already touched upon. With regard to statistical analysis, we developed a drug utilization R package, which could easily run on the data, and our output was presented via reports as well as via an interactive web application. If we go to the results, so here you see again the different databases that we used, the study period, and here you see the total number of individuals that we had could use for our study. So we had a source population of almost 42 million. If you look at use of H2 receptor antagonists, more than 1 million users, and of these users in particular, they use ranitidine. And so almost 90% of use of H2 receptor antagonist was indeed use of ranitidine. This graph shows the incidence of ranitidine use, and on the x-axis you have the calendar year, on the y-axis the incidence rate per 1,000 person, and the different colors represent the different databases. What we can, with their colors, and so dots with corresponding 95% confidence intervals, what we can first observe is that incidence of ranitidine use is low. And so for the database with the highest use, that was 11 users, new users per 1,000 persons. Of those databases with the longest follow-up, we see a decrease around the period 2005, 2010, and then either a slightly increase or results remain stable over time. We next checked incidence use by age and gender. So on the x-axis now you have age groups, and on the y-axis again, incidence rate per 1,000 persons. What you see for both females and males is that the use increases by age, reaching the highest use around 70, 79, and then it decreases again, and use was somewhat higher in females compared to males. We touched upon incidence. Now I'm showing the results for the prevalence of ranitidine use. Again, on the x-axis calendar year, y-axis now the number of users per 1,000 persons with the different databases. Here as well, prevalence was low, with the highest prevalence around 29 users per 1,000 persons. And of the databases with the longest follow-up, it quite more or less was stable from 95 until 2005, then decreased and either further decrease or catched up a little bit, depending from database by database. Then we looked for the cumulative duration of ranitidine use. And in this table, you see the use by the different databases. Here, I first present cumulative duration in days, the median, and you show that it varies from 28 to 60 median duration of exposure to ranitidine, depending by the database. We next investigated cumulative duration in specific categories recommended by the ICH. These are categories suggested when you want to explore exposure to potential carcinogenic drugs. And what you see here and what I highlighted is that use for 1 to 10 years and definitely use for more than 10 years was very low amongst all databases. Majority of patients use it for either 1 month or 1 to 12 months. Then with regard to the indication of use, first of all, what you will observe is that, so here again, the different databases, I first present whether indication is available or not. And as I said, that meant looking for specific disease code. And only for 11% of users, we had a disease code of interest in the 1 year prior to the initiation, drug initiation. If we use these patients and then categorized by the indication of interest, we see that majority used ranitidine for reason of reflux disease followed by peptic ulceration. Now, if we move to the discussion, so I think that from these data, we can indeed conclude that this was a rapid drug utilization study because it took 4.5 months from getting the protocol approval to drafting of the manuscript. But it's observational study, so we all have strengths but weaknesses as well. So the strengths is it's real life data, large numbers, so quite high external validity. And it was definitely a rapid study, which was possible because our data was mapped to the common data model, and we had an R drug utilization study package available, which could run on the data. And this also allows us to replicate this package on other databases. And we presented the data in a user-friendly way through a Shiny app. Weaknesses, as you know, as I have shown you, we had information on indication of use in 10% of new users of ranitidine. So there's definitely underreporting of indication of use, but that's because I looked for a specific disease code. There might as well be underreporting of drug use as ranitidine is also available as OTC or was available as OTC and might have been used in a hospital, which I didn't catch as well. It's prescription data, not dispensing data, and definitely not intake data. So we might have underestimated actual use or overestimated actual use. And then the cumulative duration was important, but of course, this depends on the total duration of follow-up of a patient that we have within the database. So that could be affected by that as well. So to conclude, from our data, we saw that the incidence and prevalence of ranitidine was low in the electronic healthcare databases. Use was the highest in females and increased when patients got older. Proportion of users of ranitidine for more than 10 years was less than 4%. Ranitidine is currently suspended from the market. And if we look at our data in our last study year, this implied that for approximately 100,000 individuals from these databases, an alternative treatment to ranitidine needs to be initiated if they still require treatment, of course. I want to acknowledge all authors, co-authors, and the participating research team, and of course also the EMA who provided the unconditional research funding for this project. And you find my email address, or you can contact me in case you would like to have further information. Thank you.
Wonderful. Thank you very much for the presentation, Katja. Very nice. Folks who are listening, please enter your questions in the Q&A if you have any, and I'll kick us off as well. So this actually gets to one of the limitations that you had mentioned before. I was curious, within the EHR databases, do you have a sense for how long the patients could be followed up for? You know, in a lot of US databases, we can only follow patients for a year or two, maybe 3 if we're lucky. With a CPRD, that's a different story. You know, you can follow patients indefinitely because it's a closed system. So did you have a sense for how complete the follow-up would be, the average duration?
So the average duration of follow-up depends by database as well. So similar as for the US, for each individual patient, of course, we had information on the mean follow-up, we had information on the follow-up, but there were databases with a lot, where, for instance, median follow-up would be like 6 to 7 years, we were able for some of the databases also to go back to 1992. That didn't mean that for all patients we had information from 1992 until 2019, but there were patients where we had that information. So these some of these databases definitely had a large history of data.
Gotcha. Great. Thanks. And I'm not seeing anything come through in the Q&A. I do have another question. So with respect to the information that the regulators were trying to glean from your study, it looked like you were really trying to get at, okay, how many patients are affected? What's the profile of these patients, et cetera? How do you think they used this information in their decision-making? Did it directly influence the decision to remove ranitidine from the market, or?
I think so. I think that the regulators had already made the decision to remove it from the market, irrespective of our data. But it was at the time when they were discussing this problem, this was asked. Of course, what this information provides, it gives an idea on the incidence and prevalence of ranitidine use and the alternative H2 receptor antagonists, and it gives an idea on if you remove ranitidine from the market. Of course, there are people who need treatment. What proportion of people is affected? And in a follow-up study, we are also going to investigate what happened. So it was removed from the market. To which type of drugs do they switch? And that will mainly be proton pump inhibitors, I suppose, but that's something that we want to explore and investigate in the future.
Very nice. All right, well, thank you. I think we have a little bit of time to make up on the back end here, Elaine. So I'm gonna pass it back to you.
Okay, thank you very much. Our next speaker is Dr. Donna Rivera from the U.S. Food and Drug Administration, and she's going to be discussing approaches and standardized methodologies when you're trying to compile diverse data sources and do real-world data evaluation.
Thank you, Elaine. I just want to verify that you can see my screen.
I'm seeing a general ISB background screen. Is that your first slide?
That is my first slide.
Okay, yes.
Thank you so much. Okay. Okay, so thank you so much. And thank you to ISB for the opportunity to present. I have nothing to disclose. And just of note, this work was primarily conducted when I was at the National Cancer Institute. So good afternoon. I'm Donna Rivera, the Associate Director of Pharmacoepidemiology in the Oncology Center of Excellence at the FDA, and I'm pleased to present the Friends of Cancer Research Real-World Data Pilot 2.0 Collaboration, Implementation of Standardized Methodological Approaches for Evaluation of Oncology Real-World Data, along with all of my wonderful colleagues. So we'll start with a project overview. The purpose of this project was to evaluate collective opportunities and challenges of transforming real-world data to real-world evidence for clinical effectiveness by exploring analytic definitions of potential endpoints that may be fit for assessing oncology outcomes. The objectives of this project were twofold. The first was description of demographic and clinical characteristics of patients with advanced non-small cell lung cancer receiving platinum doublet, checkpoint, or combination therapy, and additionally to evaluate and compare overall survival in the real world time to treatment discontinuation, and real world time to next treatment among patients in this cohort. We have a variety of analytic collaborators listed here as data partners. The study design was retrospective observational study, and the data was from real world data partners that included electronic health record and claims-based data within a range of settings, from community, academic, integrated delivery system networks in the United States, and the study used retrospective, patient-level, HIPAA-compliant, de-identified data. So to begin, the population cohort selected by each data partner was patients with advanced non-small cell lung cancer treated in the frontline setting with qualifying therapies. And it's important to note that the goal was to make real-world inferences, so eligibility was based broadly on understanding this population of interest. The primary criteria were cancer diagnosis, meaning evidence of advanced disease defined as stage 3B, 3C, or 4 non-small cell lung cancer at advanced or at initial diagnosis, or a diagnosis at early stage with the recurrence or progression to advanced disease, as well as the other qualifying aspect of treatment. So as you can see here, as we walk through the diagram, the eligible patient cohort with the diagnosis, then those who did not meet enrollment criteria, such as having 2 separate encounters or occasions present during the study period, or non-eligible claims-based enrollment. Additionally, qualifying treatment had to occur between 1/1/2011 and 3/31/2018. And that had to include qualifying medications, which are cisplatin, carboplatin, oxaliplatin, or nadalaplatin with pemetrexed, paclitaxel, nab-paclitaxel, or gemcitabine, as well as immunotherapies of atezolizumab, nivolumab, or pembrolizumab. And then additionally, there were other exclusions around incomplete historical treatment data, leading us to the real-world data analytic cohort of 23,102 patients. So I'm going to weave lessons learned into this presentation throughout because the real focus on this was how this project generated collaborative lessons learned. So we'll start with standardization and transparency. It was important in the study to implement a well-developed shared common research protocol a priori. We focused on determining required core common data elements from variables that were shared across sources. Sometimes variables or endpoints required alignment or definition, translation, or validation. Examples might include variables that are available in different formats, such as biomarkers, or variables requiring extraction, such as those from unstructured data. And that was largely around progression data. A standardized reporting template was utilized, which provided table and figure templates to create understanding around the intended results to be generated. And variability in data elements demands thoughtful consideration and increased transparency in a large collaboration. Each real-world data partner operationalized the common research protocol based on the characteristics of their data source, technological feasibility. And in conducting these analysis individually, they reported summary level results via the uniform reporting template due to patient privacy, technical complexity, and proprietary nature of the data sets. Developing tables within the shared research protocol allowed our groups to consider subtle differences in modeling that would not have been demonstrated without developing these in advance. Additionally, statistical analysis plans must be comprehensive with sufficient detail to reduce the risk of deviations in methods employed and characterization of variables in models or tests. This is what we are demonstrating as our table 1, and it looks a little different than a traditional because of the volume of data across data groups. We decided on a heat map presentation. Clinical and demographic characteristics were similarly distributed within treatment groups for each of the real-world data sources. Geographic coverage varied by source. You will notice in this diagram that there are differences in missingness across certain variables within the data sources, which include smoking and performance status. Overall utilization of PD-L1 and combination regimens increased over time during the study period, and the initial use of PD-L1 therapy is correspondent in 2015 with the FDA approval of the drugs. So again, thinking about lessons learned and data quality, it was deemed infeasible to aggregate or merge the data for analytic purposes among the real-world data collaborators, and instead, we sought to standardize definitions and harmonize processes. The development of each analytic data set was subject to data availability and software programming available to each partner. Each partner has established curation processes designed to evaluate the quality and completeness of their data. So as discussed before, aligning on those key covariates, and then when accepting variation, the use of sensitivity analysis to examine variance can be helpful. And in terms of quality, important to think about evaluation of these data quality aspects by the team that has deep knowledge of the curation and provenance of the data. Development of templates for quantitative evaluation of distributions, quality, and missingness, as well as quality indicators, can inform study design. Additional lessons learned in the methods aspect are around these key areas, which include missing data, a common challenge in real-world data. So including approaches for evaluating and accounting for missing data in the protocol help to maintain study integrity while minimizing bias. Imputation should be carefully considered given the potential for missingness of variables related to patient outcomes, i.e. informative missingness in real-world data, and imputation of the data or use of missing data categories in modeling has implications for study analysis and inferences. It's also important to ensure that the sample size is sufficiently large to be able to derive meaningful inferences and that the target population is well understood. And of note, statistically significant p-values do not necessarily imply clinical significance. And in totality, even with careful attention to adjustment, there are inherent selection biases and unmeasured confounding, as well as cohort effects that may not be able to be accounted for in the study. Limitations of real-world data need to be appropriately addressed in the interpretation, and transparency is valued. So discussing a bit about endpoints, and I have provided a diagram on the right-hand side of the endpoints utilized in our study, but for the purpose of time, we'll not go into how we defined each of them specifically. Commonly used endpoints in randomized controlled trials may not be practical or replicable in real-world data. Progression outcomes were not included in Pilot 2.0 because there are continual challenges with the measurement of progression. While uniform criteria allow protocol-directed establishment of progression in clinical trials through things such as RECIST, progression outcomes are not consistently captured in real-world data, and the ability for these to not be uniformly defined or captured are challenging. Therefore, real-world data sources may be better able to use outcomes such as time to treatment discontinuation and time to next treatment. And these were selected as measurable parameters to evaluate as endpoints instead of PFS. Additionally, survival outcomes such as real-world overall survival are easier to define. However, many sources have missing outcome data, which affects estimation of real-world overall survival and can limit interpretation. Linking to additional sources with more complete data may improve endpoint ascertainment. And finally, when evaluating real-world endpoints, there's a need to merge clinical applicability with analytical feasibility. One additional step in the process that may have been useful would be to evaluate the potential to share software code between groups for replicability and additional validation. I'm briefly going to show the results of the study. So as you can see here, median real-world overall survival in the platinum doublet arm ranged from 10 to 17 months across groups versus 12 to 18 months in the PDL1 arm. Directing you to the right-hand side of the slide, compared with other endpoints evaluated, real-world time to next treatment showed greater consistency of findings across sources with a significant hazard ratio across groups, indicating that non-small cell lung cancer patients had longer time on first-line treatment with PDL1 than platinum doublet in this setting. This also suggests that time to next treatment may be less susceptible than other endpoints to the impact of data variations that currently exist in real-world data sources, but also highlights the importance of sample size in the parameter estimates. Additionally, whether time to next treatment could be appropriate as an endpoint in comparative real-world data analysis for a regulatory objective would require additional consideration, as this endpoint does not strictly measure efficacy. Further validation would be necessary as it has not been evaluated in the clinical trial setting. However, it could be considered alongside other measures in a pragmatic prospective design. And lastly, I'd like to touch on limitations because I think it's important for transparency that there were some limitations to this study. It is unknown to what extent patients are represented in more than one data source, duplication. There was, as I mentioned, methodologic variance, missingness in certain types of data, and the potential for systematic information biases. as well as the control of potential confounding in this study being limited by the need to implement a uniform analysis across data partners to achieve the broader research goal. So in conclusion, this study illustrates the power of multi-stakeholder collaboration to identify both the challenge and the importance of methodological rigor in real-world data efforts to support the generation of high-quality real-world data. We established several processes, and the heterogeneity across data sources exemplifies the challenge in interpretation of real-world evidence in the context of traditional randomized controlled trials, especially in any direct comparative or emulative efforts. Future research on methods for standardization of an approach to categorize data and establish objective measures of data quality that incorporate pertinent real-world data assessments is needed. And very briefly, the real-world pilots led by Friends of Cancer Research, Pilot 1.0 was a feasibility study, and the publication is available. The study I just presented on the collaborative performance of real-world endpoints is part of Pilot 2.0, along with Part 2, which evaluated internal consistency by applying randomized controlled trial eligibility criteria. And lastly, real-world response. Establishing a definition of real-world response in real-world data is important, and there is a new pilot project being launched to evaluate the feasibility and reliability of collecting response measures across data sources for methodological evaluation. With that, I would like to especially thank my excellent forward-thinking colleagues at the Friends of Cancer Research and the variety of healthcare organizations and collaborators, as well as federal collaborators at FDA and NCI that contributed to this project. Thank you so much for your time and attention.
Thank you very much. That was really fascinating to hear, and I think it underscores the attention to detail and rigor at every step of the way of doing these kinds of analyses. So I think we have time for one question. My question is, knowing that you had so many collaborators, one thing you said is it takes a team-based approach of having people with deep knowledge of the data provenance, and people may not know, well, who do I have on my team? So since many of us here are pharmacoepidemiologists, what does that mean? Who do we need to have on our research team to make these kinds of initiatives work?
I think that is such a great question. And I'm so glad you asked. We've actually thought a lot about this in the team science context. So I think in every real world data collaboration, much of the work we're doing is clinical. So I think the representation of clinically trained individuals, additionally, people that are familiar, trained in data science, the aspects, deep knowledge of the data, methodologists such as pharmacoepidemiologists, alongside statisticians. I think our statistical colleagues have a lot of deep knowledge in a lot of the analytic methods that need to be applied. And also, I think important for us to think about patients. Often, they have valuable aspects of whether it's patient-reported outcome data or other types of data. So I think this really is trying to bring everybody on the team and all their respective expertise together to truly advance the science of real-world data.
Thank you very much. I'll pass it on to my colleague for the next presentation.
Thank you, Elaine. Thank you, Donna. Nice presentation. Up next, we have Lin Wang, who's a PhD candidate at the Department of Epidemiology at the Johns Hopkins University Bloomberg School of Public Health, and she'll be presenting on cost-effectiveness analysis of prostate cancer treatments. And Lynn, you're currently on. Yes, we can. We can hear you too. Thank you.
Thank you very much. And welcome everyone. I'll present cost effectiveness of systemic treatment for metastatic castration-sensitive prostate cancer and economic evaluation based on network meta-analysis. And here's the disclosure. So for metastatic castration-sensitive prostate cancer, continuous androgen deprivation therapy represented the standard of care from 1941 until 2015, when randomized controlled trials showed that docetaxel improves overall survival when added to androgen deprivation therapy. Following docetaxel, abiraterone acetate, apalutamide, and enzalutamide were subsequently approved for this indication, based on randomized controlled trials showing that their addition to androgen deprivation therapy improves survival outcomes over androgen deprivation therapy alone. The availability of these drugs has improved prostate cancer outlook. In the meantime, the multitude of treatment options has complicated decision-making, as there is a lack of clarity in the comparative effectiveness and safety compounded by a wide range of drug costs. The unclear comparative effectiveness is due to the lack of head-to-head trials and is reflected by the hesitance of clinical guidelines to commend one drug over the others. If we look at the National Comprehensive Cancer Network guideline for metastatic castration-sensitive prostate cancer, all 4 treatments are listed as category 1 recommendation, which means based on high-level evidence, there is uniform consensus that these interventions are appropriate. However, the drug costs vary widely. For a patient to complete all planned courses of treatment, the drug acquisition costs range from around $300 for docetaxel to about $260,000 for apalutamide. This is because docetaxel and abiraterone are generically available, while enzalutamide and apalutamide are patented for this indication until 2027, 2033, respectively. So the objective of this research is to compare the cost effectiveness of systemic treatments for metastatic castration-sensitive prostate cancer through an economic evaluation based on a Bayesian network meta-analysis of clinical trial data. The network meta-analysis part of this research has been published in JAMA Oncology. In brief, we identified 7 trials with 7,287 patients comparing 6 treatments with a follow-up duration ranging from 14 to 84 months. Allowing hazard ratio to change over time, we found that abiraterone and apalutamide were associated with the largest and most consistent survival benefits. We'll focus on the economic evaluation part for today's talk, which has been accepted in Value in Health. Based on the network meta-analysis, we assess the cost effectiveness of these drugs over a lifetime horizon from the US health sector perspective. We assessed 5 treatment strategies: docetaxel, abiraterone acetate, apalutamide, and enzalutamide, plus androgen deprivation therapy versus androgen deprivation therapy alone. The dose regimens are as listed. We used the partition survival model to model disease progression and treatment effectiveness in a cohort of simulated metastatic castration-sensitive prostate cancer patients as they experienced 3 different health states: pre-progression, post-progression, and death. The partition survival model is a commonly used decision analytical approach for appraisals of interventions for advanced or metastatic cancers. To model survivals, we first digitalized survival curves of androgen deprivation therapy arm in randomized controlled trials, combined them with network meta-analysis and to derive the parametric survival curve for androgen deprivation therapy arm. Then we calibrate the long-term overall survival curve for androgen deprivation therapy alone arm with SEER data to improve the survival extrapolation beyond trial duration. We then applied time-varying hazard ratios derived from the network meta-analysis to generate survival curves for androgen deprivation therapy plus chemohormonal therapies. We evaluated costs, including treatment-specific costs, health state costs, and adverse event costs. For treatment-specific costs, we included drug acquisition costs and administration costs. The drug acquisition costs were sourced from the Department of Veteran Affairs' latest federal supply schedule contract to reflect actual drug costs to federal agencies after discounts and rebates. The administration costs were sourced from Medicare physician fee schedule. We derived health state costs from market scan database. We obtained adverse event costs from AHRQ HCUP project. The probabilities of adverse events were informed by FDA drug labels. We included adverse events rated as grade 3 to 4 in severity and occurred in 0.5%, greater than 0.5% of the patient population, considering their medical significance as well as cost and utility implications. We measured the health-related quality of life by a utility score on a scale of 0 to 1, where 0 indicates death and 1 indicates perfect health. Utility values were sourced from published literature that have used proper preference-based methods such as EQ5D. We evaluated health outcomes in QALYs and measured cost-effectiveness by incremental cost-effectiveness ratio, ICER, which is the incremental costs incurred divided by the incremental QALYs, quality-adjusted life years gained, comparing two treatment alternatives. In the base case, the model parameters took their mean value. In the sensitivity analysis, model parameters were sampled from their respective distributions. Here we tabulate the results where treatments in the first column are ordered by increasing QALYs in the second column. If we look at the last column, ICER, enzalutamide was dominated by docetaxel because of its higher cost for the same QALYs. Docetaxel had an ICER of 42K per QALY gained over the androgen deprivation therapy alone. Abiraterone acetate had an ICER of around 59K per QALY gained over docetaxel. And apalutamide had an ICER of nearly 2 million per QALY gained over abiraterone acetate. This is because with the mean of 0.25 QALY gain, it incurred an additional 530K cost. Here we show the probability of a treatment being the most cost-effectiveness one across all treatments against a range of willingness to pay thresholds. We can see that at a willingness to pay below 50K per QALY, docetaxel is likely the most cost-effectiveness treatment, while at any willingness to pay threshold between 50K and 200K per QALY, abiraterone acetate is likely the most cost-effectiveness treatment. Thank you for listening.
Great, thank you very much. A very interesting presentation. I'll kick it off with a question. So with respect to your choice of data sources, I was curious, you had used the MarketScan databases for the health state costs and some HCUP databases to cost out the adverse events. Can you speak to why the HCUP data seemed like a better source than the MarketScan data for AEs versus the health states?
It is mainly for the convenience purpose because HCUP, it has around it represents 25% of the US population, and it has both patients from commercial insurance and also public insurance. So it's very representative, and you can use DRG ICD-10 to identify the hospitalization costs of any adverse events.
And it was so did you use the nationwide inpatient sample then? Or the national inpatient sample is what they're calling it now? Okay, got it. Interesting, great. And then from the lifetime perspective, the model is developed from the lifetime perspective. I'm curious if you have a sense of what the expected duration of overall survival is in an individual who would be appropriate for these treatments. Like how long would they be expected to live on average?
I need to pull up my, it will be included in the Value in Health paper. I have extrapolated overall survival curve for their life expectancy under different treatment strategies.
Got it, great. And then can you expand on what the health state costs comprised? Like what did that mean and how did you go about estimating those?
Yeah, so we constructed a cohort of patients who had metastatic castration-sensitive prostate cancer based on their ICD-10 code and ICD-9 code for patients who were recruited earlier in time. And then we identify when their disease become castration resistant, also based on ICD-10 and ICD-9 code. And then during that period when their cancer is castration sensitive, we identified both inpatient costs, outpatient costs, and we just use that to net the treatment specific costs. So the drugs that we're interested in this cost effectiveness analysis.
Okay, great. Thank you. I'm not seeing any other questions in the Q&A. Just one other question. So with respect to the castration resistance and castration sensitive, so they actually have ICD codes that indicate that clinical feature of prostate cancer, or did you use a proxy like the fact that they didn't get surgery just was indicative of that type of cancer?
Yeah, there's a specific ICD-10 code for that.
Okay, yeah, that most of the ICD-10 codes often don't have that nice level of clinical detail in cancers, like for, I don't know, breast cancer, et cetera. It'd be nice if they had that for all of them.
Yeah, sometimes we need to develop an algorithm for the cancers you mentioned. Yeah.
Okay, great. Well, thank you very much again. Great presentation, and I'm going to pass it back to Elaine.
Thank you very much, Stephen. So our next presenter is Dr. Lu Chen, who's a senior data scientist at Genentech, and she'll be talking about the impact of rising use of telemedicine, and in this case, how things like a pandemic forced innovation in how we think about our real world data. So I'll turn it over to you now.
Thank you. Can you guys see my slides okay?
We can see your slides and hear you fine. Thank you.
Great, thank you. So thank you for the opportunity to share our results here. This is our disclosure. So this study is a collaboration between Genentech, that's where I come from, and our authors from Flatiron Data, that's the data we used. So this study really aimed to understand the patterns of telemedicine use in cancer patients during this COVID pandemic. This question became important because several factors may have influenced the role of telemedicine in oncology care during this special time period. First, telemedicine can be used to mitigate the spread of COVID, given its ability to deliver care remotely. Second, on March 17, 2020, the CMS expanded the reimbursement of telemedicine services to all beneficiaries, which was previously limited to non-urban areas or areas of physician shortage. So these factors may have impacted the use of telemedicine in cancer care, which may also have implications on study using EHR data. For example, shall we incorporate telemedicine visits in estimating follow-up time and care gaps in studies of patients during this time period? So to answer this question, we used Flatiron data to construct 4 cancer cohorts. They are patients with metastatic breast cancer, DLBCL, multiple myeloma, and advanced non-small cell lung cancer with respective diagnosis after January 1st, 2011, who were still alive by March 1st, 2020. Then we follow them from March 1st to the end of 2020, or death, whichever came first. Telemedicine was defined as billable remote evaluations and management. In this database, traditional visits or in-person visits was defined by the presence of a medication administration, a lab test, or a vital sign record. We first examined monthly telemedicine use in patients and patients' characteristic by telemedicine use in the entire cohort. Then we took a deeper dive into 2 cancer types, DLBCL, where we saw a relatively higher telemedicine use, and advanced non-small cell lung cancer, where we had the largest sample size. So within these two cancers, we then created patient subgroups according to where they were in terms of treatment. These were first, those who were newly diagnosed in the beginning months of the pandemic. Second, those who initiated first-line treatment among those who were newly diagnosed. And the third group were those who had started treatment before the pandemic and continuing their first-line treatment into the pandemic months. And lastly, those who are on established treatment since 2019. So in each of these subgroups, we calculated follow-up time and proportion patients with care gaps in two ways, with telemedicine and without telemedicine. Follow-up time was the difference between the last visit and the later of March 1st or the diagnosis. And the care gaps were those, the period of 60 days or longer without a visit. So as shown in this graph, we observed monthly telemedicine use peaked in April and May across all cancers during last year. The use flattened during the summertime and went up again in November and December. This trend tracks with COVID waves nationwide. We also observed some regional differences in the uptake of telemedicine following the spread of COVID across the US. So here we're showing DLBCL as an example, each bar representing monthly telemedicine use for a region. You can see telemedicine was adopted more rapidly in the Northeast region in April and May. In the later half of 2020, telemedicine was more frequently used in the West region. So DLBCL is just one example, and all other cancer types follow the same pattern. We also observed that telemedicine was used mostly as a supplementary care. This graph shows the proportion patients by visit type. Telemedicine only, which is the red tip on the top, people only using the traditional in-person visits, that's the gray bar in the middle, and people who used both type of visits, that's the blue sections at the bottom. As you can see, patients who were exclusively using telemedicine remained smaller than 5% across all cancers during the entire period. We also compared patients' characteristics by the telemedicine use. So we observed some differences by care sites and treatment as shown at the bottom rows. Specifically, telemedicine users were more likely to receive care from academic sites and were slightly more likely to be on oral agents only. Otherwise, there was no major difference by sex or disease stage. I'd like to point out here that in this particular database, lung cancer was advanced lung cancer patients. They only capture patients who had advanced disease. So it's possible that the initial stage was less relevant at this time point, and hence we didn't see a difference by stage. And in terms of how telemedicine impacts on EHR studies, we examined two measures, follow-up time and care gaps. So these were, as I mentioned before, were calculated with or without telemedicine in patients with DLBCL and advanced non-small cell lung cancer. We did so separately in subgroups of patients defined by care stages. So across subgroups, there's no difference in the length of follow-up time calculating with or without telemedicine. And as shown in this graph, the proportion patients with a care gap were also very similar, calculating either way across cancer types, across subgroups. So these findings suggest limited potential for misclassification in follow-up time or care gaps as patients adopt telemedicine. So to conclude, telemedicine use in cancer care was used in cancer care to complement traditional care during the pandemic, with its role varying by disease type and specific patient characteristics. Telemedicine use peaked in April and May across cancer types, but patients exclusively using this service remained small. While the short-term impact of including telemedicine in EHR studies was limited, we do recommend more research look at longer-term measures and more complex measures. that's all I have for this study. Thank you.
Thank you very much. This is so timely in terms of seeing the real world data of the real world data, so to speak. I was really struck by the 4% number and estimate, and I wonder your thoughts on how generalizable you might think that is across other treatment areas, if you know or others are reporting it. And what comes to mind is I might imagine its effect, it relates to the kind of condition being treated, in this case, cancer care, and also maybe the generation and people's comfort in seeking care tele-wise. I think of my grown adult kids who are much more comfortable in telemedicine than maybe my grandparents, so to speak. Your thoughts, is what you're seeing in other therapeutic areas similar in that 4% range or varying?
Thank you for the question, Elaine. I haven't looked at other therapeutic areas, but this was a surprising finding to us as well, because at first we were worried that telemedicine may take up more patients, and that's why including or excluding televisits may have a bigger impact on our studies using EHR data. So for our researchers, I think this is a reassurance that telemedicine was small. So whatever we did before may still hold in this special time period. When we're looking, we didn't show in the data here, but we did look at difference by age in terms of who adopts telemedicine, and we didn't see a big difference. Granted, most of people here we're seeing are cancer patients and they're older. Yeah, so that's what we found here.
Yeah, so maybe an area of further study to really know, as you're saying, what impact. So, and I know at least in the US, there's a lot of heated discussion around whether or not telemedicine is going to stay in certain areas or it might just grow. So your advice then on should this be something that we all standardly incorporate in when we're considering potential confounding or biases in our analyses?
I'd like to speak in two folds. For now, with this limited data, we don't see a strong confounding by including or excluding televisits for these special time periods. Granted, if you're comparing historic cohorts to these time periods, there may be some differences because telemedicine was not as rigorously recorded or not recorded at all in the previous EHR databases. So that's something people have to look into in more details. And here we're only looking at two measures, which is based on the visits only. But a question remains is what being captured during the telemedicine, was that at the same level of details as before? For example, medication use or even vital signs. Do people self-report or just the quality of the data compromised during telemedicine? I think people have to do more research to look at that.
Yeah. And we have our first question from the chat of the group, so I'm going to quickly read it. So thank you, Benjamin. He's asking, are you or others considering differences in the clinical outcomes among people who receive telemedicine versus standard visit? You know, something to continue to monitor.
I think that's a very interesting question. And of course, you know, we were thinking about this when we started the analysis, but we didn't do that for two reasons. One is the follow-up time is really short. It only happened last year. So if you were to see something, it's unlikely to see it right away. And the other thing, as I mentioned, maybe in the slides here, we do see that slightly people who are using telemedicine only were those on oral agents. So there is indication by confounding who can just do telemedicine, probably are those people who are less severe. So it's hard to tease apart if it's the effect from telemedicine or it's because people who are less severe can do telemedicine.
Yeah, still more to understand in this area that I'm sure will continue to evolve post-pandemic. So for our last talk, thank you very much. And back to you, Stephen, for our last presenter.
Great, thanks. Interesting presentation, Lou. As somebody who does a lot of research in the area of surgery, the pandemic has had a major impact in all the research that we've done as well. So it's interesting to see how it continues to pan out. Our next presenter is Dr. Mohammed Movahedi. He's an assistant professor at the Institute of Health Policy, Management and Evaluation at the University of Toronto. And he's going to be presenting on physician and patient reported outcomes for tofacitinib and TNF inhibitors in a Canadian RA registry.
Okay, are you able to see my slides?
Yes, we can see them and hear you well. Thank you.
Okay, thank you so much, and thanks for giving me this opportunity to present the results of a study we conducted recently to compare the physician and patient-reported effectiveness outcome between TNF inhibitors and tofacitinib using our registry as a real-world data. I'm pleased to present this on behalf of my colleagues. And this is our disclosures. I'd like to start the background for those who are not familiar with the anti-rheumatic medication. Tofacitinib is an oral small molecule drug used for treatment of rheumatoid arthritis and usually is prescribed as alone or with methotrexate. So since approval in Canada in 2014 and in 2012 in the United States, TOFA has been a good replacement and has been an area of interest for the clinician to be used in place of biologic because the way of administration, which is oral, and also because of some cost. So there are lots of interest and questions around comparability of tofacitinib in regarding of effectiveness, adverse events, and also sustainability. In my presentation 2 days ago, I presented the sustainability of the tofacitinib versus TNF inhibitors, and in that presentation, we showed there is no significant difference between these two medications. Now I'm just focusing on evaluation of physician and patient-reported effectiveness between these two treatment groups. A little bit about our data registry. Ontario Best Practice Research Initiative is a multi-center registry across Ontario, over 70 rheumatologists involved for data collection. Also, we are collecting data from patients by doing telephone interview every 6 months. For telephone interview, we usually collect the social demographic, physical function of the patients, including some patients reported outcome, also comorbidity, as well as non-rheumatoid arthritis medication. And patients are allowed to see their rheumatologist as per routine care. And in each visit, we usually collect the data from the physician activity, like disease activity, like clinical disease intakes, including swelling joint, tender joint, physician global assessment, as well as comorbidity, as more importantly, start and stop of anti-rheumatoid medications. So for eligibility criteria to be enrolled in the OBRI, patients should be more than 16 years at time of diagnosis and more than 18 years at enrollment, and must have the rheumatologist confirm RA diagnosis and at least one swelling joint count. For this particular analysis, we designed a study as a retrospective cohort study. Those patients who enrolled in OBRI and initiated their TOFA or TNF inhibitors between June 2014 and December 2019 were included. As I mentioned earlier, 2014 was chosen because TOFA approval date was in Canada. For TNF inhibitors, we included individual TNF inhibitors, adalimumab, certolizumab, etanercept, belimumab, infliximab, and biosimilar for infliximab and etanercept. So in terms of inclusion and exclusion criteria, patients were required to have physician and patient-reported effectiveness data available at treatment initiation and a 6-month follow-up plus minus 2-month window. These included clinical disease activity index, CDI, rheumatoid arthritis disease activity index, which is collected by patient interview, health assessment questionnaire disability index, a sleep problem, and anxiety and depression scores. As we know, for real-world data, we have 2 challenges. The first challenge is dealing with the confounding by indication. I believe majority of people and attending here are familiar with the confounding by indication. Just very briefly, patients with starting with the disease, different disease profile, including disease activity or comorbidity, might get a different chance of prescribing a particular medication versus the other medications. So we must be sure to balance between these two, and because it's the nature of observational study, we usually, and with lack of the randomization, we have to address this problem. And in this particular analysis, we use the propensity score for those covariate with more than 10% standardized difference between two treatment groups, which is tofacitinib versus TNF inhibitors. We use two most common approach of estimation for propensity score, stratification, and also stabilized inverse probability of treatment weight methods. For the main analysis, because this is the repeated repeated analysis, repeated measures as a longitudinal study, we use the general linear mixed model to compare these two patient report outcome and physician effectiveness between two treatment group. And also, again, another challenge for observational study is the missing data, particularly for those main covariates, and we apply multiple imputation using imputed chain equation method. In terms of the results, this table just summarizes the main variables as an illustration of TNF inhibitors or tofacitinib. Overall, 429 patients included, 226 as a TNF inhibitors and 193 as a tofa users. The main important difference was for the age. Patients in tofa group was older. They more likely to be female, but it was not significant, as just showing by just the simple p-value and chi-square p-value. The disease duration, patients in tofa group was longer disease duration, had longer disease duration. The health insurance coverage somehow was higher for tofa group. ACPA, which is one of the biomarker for rheumatoid arthritis patient, was less likely to be present in tofa group. The mean of CRP was less, and the most important part was because tofa users usually use and prescribe after failure of the prior use of biologic. The number of prior biologic was in tofa group was more higher than TNF inhibitors, 1.4 versus 0.3. And the concomitant methotrexate was almost the same. And more interestingly, cardiovascular disease prevalence was higher in tofa group compared with the TNF inhibitors. However, the total number, the mean number of comorbidity number was almost the same. And these covariate with the more than 10% standardized difference was used to calculate the propensity score, age, health insurance coverage, RA disease duration, hypertension, CVD, anxiety, depression score, a sleep problem, positive rheumatoid factor, positive ACPA, C-reactive protein, as I mentioned, HAC, first use of treatment, number of prior biologic use, and steroid concomitant use. In this table, just a descriptive table just shows the effectiveness and functional improvement at 6 months followed by treatment. As we could see here, the CDAI or LDA remission, which is measured as a less than 10 as a total score of 0 to 76 as a scale, we could see 33% in tofa users and was achieved remission versus 36.7% in TNF inhibitors. However, when you look at the other variables as like PRO mean change from the baseline at 6 months, you could see there is no significant differences in the mean of the change of mean between two groups. All are not significant descriptively. However, because we didn't adjust for any covariate in this table, we did try to have that adjustment for propensity score, which brings us to the table 3. You could see we have looked at compared TNF inhibitors versus tofa. The first column is just for the remission of CDAI, LDA or remission CDAI, or DAS, which is another composite measure for disease activity for rheumatoid arthritis. For unadjusted, we could see a trend in favor of TNF inhibitors. However, the p-value was not significant. When we did complete case analysis, which means we didn't apply any multiple imputation, the trend and the odds ratio was changed, but however, again, it wasn't significant, even though after applying multiple imputation and adjusting for propensity score across 20 different data set, imputated data set, you could see here again the significant level is not, the p-value is not significant, 0.85 for CDAI LD remission and 0.66 for DAS remission for using a stratification, and almost the same for using the weight approach for propensity score adjustment. And this table also shows the just coefficient, because the variable we have used as an outcome for patient report outcome all was continuous variable. So the estimates is presented by coefficient. Again, the same scenario on adjusted complete case analysis and adjusted for propensity score using multiple imputed data. Again, we could see for across the different measures of the patient report income outcome, we could see again, it doesn't matter on unadjusted or adjusted, you can't see any significant level of difference between achieving this patient report outcome and decreasing the level of the fatigue, HAQ, red-eye, global pain, morning stiffness, or sleep problem. We could see there are some trend in favor of TNF inhibitors. For example, if you look at the HAQ, you could see the coefficients unadjusted is -20. But when we look at the complicated analysis and the multiple imputed adjusting for propensity score, this difference and these coefficients get to almost nil. That means there is no significant difference between these two. And the same you could see for fatigue, for red-eye, and global pain, as I mentioned, morning stiffness and sleep problem. And that brings us to the conclusion. In this real-world data set, we found that physician and patient-reported effectiveness outcome are similar in TNF and TOFA group 6 months after initiation of treatment in patients with rheumatoid arthritis. However, as you might have mentioned, the number is not high, it's just about 500 patients. So one of the concern about the study power and analysis power to detect a significant level of difference. So what we are going to do is merging our data with another RA registry in Canada to increase the sample size and do and have the enough analysis power to detect any possible significance level difference. Thank you so much, and happy to answer any question you have.
Okay, great, thank you. Very nice presentation. So no questions in the Q&A. My first question actually has to do with that last point that you made around statistical power. I'm curious, if you go back to your results table that had the, it was the second adjusted results table with the various different PROs. Sometimes
this one?
I think table 4, yes, thank you. I'm not quite as familiar with some like the interpretation of the coefficient. Would you say that sometimes when we do an analysis and if it's underpowered, we might have an effect size that looks clinically meaningful, but our confidence interval is really wide. And then sometimes you have an effect size that's both statistically insignificant and clinically insignificant, which it sort of makes you feel a little more comfortable that the finding isn't just underpowered. What magnitude would a 0.41 change in a fatigue scale from 0 to 10? Can that be directly interpreted like a linear regression, or would you say these are clinically significant or quite small?
Yeah, this is just shows us the linear regression because the outcome is continuous. So we can't see, it's not just like binomial or binary outcome, which I showed in the table 3. But
Right.
however, it just says compare with TNF inhibitors, compare with the, TOFA has got over the time, the score of fatigue is getting better by 0.041.
Okay, so it's like
So,
half a point on a 0 to 10 scale.
yes, that
Almost.
means that the fatigue score has get lower for TNF inhibitors versus
Right.
TOFA. However, in terms of clinical interpretation, because we have lots of our investigators, our rheumatologists, when we present this result, they show, well, this is not going to be a big change and clinical meaningful for to see there is some against or favor for any of these medications.
Got it. Cool, great. The second question that I had was the follow-up period is a 6-month follow-up, and rheumatoid arthritis is sort of a lifetime disease. Do you expect that these results would change over time? And do you have any thoughts about whether patients, and I'm not familiar with the literature at this, I used to work in RA a little bit years ago, but do you expect any difference in adherence to an oral medication versus a TNF, like an infused or self-injected medication over time? Do you think that would influence results over a longer duration follow-up?
Yeah, this is very good question, actually. And the rationale for doing some kind of this analysis is just, as I mentioned it in my introduction and in my background, TOFA is oral, is user-friendly, and is going to be prescribed by patient, and it can affect the adherence as long as we can prove there is no significant difference in terms of effectiveness, and more importantly, in terms of the adverse events. It was, I think, in the last 6 months, there was some literature review and some warning from the pharmaceutical company in terms of tofacitinib might have some more adverse events compared with the TNF inhibitors, particularly for patients over 60 years old and with some background of cardiovascular disease. So if that issue going to be not a big issue, then I think, yes, the adherence in the long term should be in favor of TOFA versus TNF inhibitors.
Gotcha, great. Very nice. And do you think that, so if I recall correctly, and this was back in 2012, I think when TOFA came on the scene in the US, it had a little bit of a rough uptake. Like it wasn't as, people weren't using it as widely as they expected. Do you think that findings like this will influence clinical practice in Canada? Ultimately?
Yeah, right now, again, back to my point, right now, investigators and clinicians in Canada are half-half. Some are in favor of still using TNF inhibitors, and some, because of the way of the administration route of TOFA, are in for tofacitinib. However, as I mentioned earlier, all this decision depends on actually the side effects and the effectiveness, which is going to be shown by real world data. And there are some few real data world, including US Corona Registry. They have already shown similar results with our current results. There is no significant difference in the pain between tofacitinib and TNF inhibitors, and also for the proportion of remission, CDAI remission or LDA between TOFA and TNF inhibitors, and this is consistent with our results. That means we shouldn't worry about the effectiveness. However, the question about adverse events is under debate, and investigators just looking the literature and watching the literature to see what's the update about this.
Great. Thank you so much. This is a great example. The RA space is a great example of a real world evidence that really needs patient and provider reported outcomes in the spirit of this session. This is great. So I don't think there are any further questions. So I'd like to thank all of the attendees as well as the presenters and Elaine and our colleague James for helping us get set up. And I hope everybody has a wonderful rest of your day.
Thank you.
Thank you very much.
Bye.
Bye all.
You.
Bye-bye. Great discussion.